in its native state at 3.8 angstrom resolution, which is not bad under the circumstances. And we found that there is something stuck to it. So it is in the process of chewing up a substrate. Proteasome, yeah. Yes. Yes. Yes, we were able to. So if we, so once we identified that this may be one of the particles, then what we can do is we can kind of use a picking process. And there are different kinds of picking programs, and many of them are sort of guided by AI. That in different orientations, this is what the particle looks like. So along with noise, can we look through the data set to enrich the data set for only these particles? So we can separate out these things in 2D. So
is the relative concentration, I mean, is that equals to the guide to say, okay, this is a valid member of my grid? Or is it just an anomalous presence that I?
No. So in fact, in the first data sets, we got things which were probably present in larger quantities. The structures that we solved of the proteasome and some other complexes that I'll show were present in relatively larger amounts from because they are being secreted. So what defines it? So there's a way of working out resolution in cryo-EM, which is called FSC correlation. So you have two half maps, and we just go through. So this is fairly standard. So you just go through different resolution ranges to see where your correlation between the half maps falls below a certain number. So the gold standard is 0.143, and based on that, we have this value. However, this is very important, and one speaker was presenting this. This is the global resolution. Yes.
The largest particles
yes.
You have larger particles. This is the largest.
This is the largest. Yes. So that is why, and because of the distinctive shape, we just went to that first. Yes. Yeah, I'll come to that. So I should also mention that 3.8 angstrom is the global resolution. So this is something that I think a speaker was mentioning. So 3.8 does not mean 3.8 throughout the particle. So there is distinction in local resolution, and the resolution right here where the substrate is was actually fairly low. So while we think we know what the substrate may be, we don't really have very defined side chain density. So we can't say with confidence what it could be that the proteasome is chewing up. But to answer the question, so this is essentially what proteasome looks like. There are 4 rings. So the central 2 rings are beta rings, and then we have 2 alpha rings, the cis alpha and the trans alpha. And we did quite a lot of comparison with the mammalian proteasome as well as malarial parasite proteasomes. The recombinant structures are solved. And what we find is in comparison with the malarial parasite proteasome, what we find is that, let me just go here. So essentially what we find is that the central cavity in the cis alpha ring is opened, and that is because it's accommodating the substrate. So this is the model which we have fitted onto the map. And what we see is that there is radial outward movement of the helices from the cis alpha ring. And also these are the loops which are involved in substrate association site binding. So essentially the magenta here is our structure, and the blue is the recombinant malarial parasite proteasome. So we see significant shifts in the way that the loops sort of move outwards, and the central region where the substrate is actually supposed to bind changes its conformation quite significantly, because this is something which is actively chewing up substrate. However, we could not say what the substrate is because of the variation in terms of resolution. Now, proteasome, another 2 other things that we found, this is also related to the proteasome, just the cis alpha ring in the intermediate state, not fully formed and fully formed. So here you see that the subunits are all packed together, and here the subunits are a little bit further away from each other. So this could be during association or dissociation, but this was also secreted by the parasite. Now, another relatively smaller complex that we could also find which had a distinctive shape was a complex that looked like this. This is again a 2D. We did not know what this is at all. We solved a structure. The resolution was stuck at around 3.4 or something. And then we realized that this looks like it has symmetry. So it has D3 symmetry, which is two C3 components sort of stuck on top of each other. So we applied D3 symmetry, and immediately the resolution got better. And, even at 3.4, we could see, quite a lot of detail. So the question was, what is this complex? So here we took the help from several different methods. One is cryo-ID, which is a method developed by Hongzhu at UCLA, and then Model Angelo, which is a fitting program developed by Shores versus Lam. So the idea here is that the density is good. Ambiguous assignment of, let's say, 6 to 7 amino acid stretches, at least 20 of those, then we can utilize that to work out what this protein is, right? And here we also have help from mass spectrometry, because this entire sample is subjected to mass spectrometry. So what is there which may have a D3 symmetry and is a part of the malarial parasite genome, and then also symmetry, which helped us to identify what could be this protein complex. No. So we have the sample, which we are getting from the malarial parasite surface. So we are doing mass spec and cryo-EM in parallel. From the grid? No. So it's the same sample that you put it on the grid and you do mass spec of. But mass spec is, it eventually as an enzyme called M17-leucyl aminopeptidase. This is a very crucial enzyme for the malarial parasite. It is required for a sort of processing of protein within RBCs at the merozoite stage, and this is also secreted. And if this gene is removed, so it is one of the it's a neutral aminopeptidase. And if this is removed from the malarial parasite genome, then the parasite doesn't survive. So it really requires this for survival. And this is also a target for various types of inhibitors or drug candidates which have been developed. So this is what the native structure looks like, and I'll come to that. And another similar structure, which appeared to have dodecameric symmetry, again looked something that we can potentially pick out of the 2D or from the raw data. And we were able to, again, apply symmetry, utilize cryo-ID, utilize Model Angelo, and some pre-knowledge from mass spec to identify this as M18 aspartyl aminopeptidase. So this is also an aminopeptidase which is required by the malarial parasite for survival. Now, for both of these, recombinant protein structures are available, meaning these enzymes have been expressed in equal and their structures have been solved. So the question is, it is interesting that we are able to work backwards and identify what this protein. Here is the recombinant M17-leucyl aminopeptidase structure, which was published, I think, in 2014. what was very surprising to the authors was that while similar enzymes in the malarial parasite have very obvious channels for substrate entry and product release, this did not seem to have much of a control mechanism, so to speak. So what would control the entry of the substrates in the active sites and what would control their release from the active site was not very obvious from the crystal structure of the recombinant protein. But what the authors had mentioned was that there is a partial loop. So there's a loop that kind of covers the entry to the active site because but the densities for the amino acids are missing. So the loop was quite significantly disordered. But they suggested that the loops could be the ones that are controlling the entry of the substrate. And they were very clear that the entry of the substrate must be controlled because this is a enzyme which can only process small peptides. So it can't just take anything. It has to be quite specific in the way that what it can handle. So here, what we found in our native structure from the malarial parasite, while the active site stays more or less the same in the native structure versus the recombinant structure, what we do have is the density for the whole loop. So the loop is some 246 to 260 amino acids, and we have side chain densities and main chain densities for the whole loop. So in the native structure, this is ordered, plus this is a zinc metalloprotease. So it requires 2 zinc ions for its activity. And in the recombinant structure, there was 1 tightly bound zinc, but the other was loose. So the occupancy was very low. And again, what the authors predicted is that this loose zinc may or may not be needed for catalysis. And the inhibitor design strategy is based on one metal binding site not being very stringent. What we find is that both zinc ions are strongly bound in the native structure that we have isolated from the parasite. Okay, so going into what we can get because we are now seeing the loop density, what can we say about the process of control of substrate binding to these enzymes? So I should mention that we solved the first structure without using any symmetry. So C1 means without utilization of any symmetry. So there is no averaging of the density. And here the global resolution is 3.4. And when we impose D3 symmetry, so this imposes averaging, and the global resolution came down to 2.75. However, in both the cases, we saw what the active site kind of looked like. And so there are 6 active sites because there are 6 different moieties that make up the D3 structure. So in the context of the D3, what we find is that we find about an intermediate size, and I'll come to what closed, open, and intermediate means. One intermediate size sort of opening corresponding to the active site. So the loop has moved away from the active site and the active site is open. However, in case of C1, where there is no density averaging, what we find is in case of some of the monomers, the loop completely closes the active site. So this is one example. But in some of the other monomers, we see partially, the loop is partially covering the active site, or it is completely away from the active site. So there's quite a lot of variation in the way that the loop is appearing to control access to the substrate binding site. So we took this native structure and we carried out 300 nanosecond simulations with 3 repetitions to try to see the positioning of the loops in all of the monomers. So what we find is that if we just do a principal component analysis of the loop, C alpha, kind of their Cartesian coordinates, what we end up with is, and if we measure the minimum distance of the loop from the active site, what we end up with is about 33.4% of the time the loop appears to close the active site. About, again, 33% of the time it is in an intermediate stage where it's slightly moved away, and then completely open, again, about 34% of the time. So there appears to be quite a lot of interconversion between these states. And the interconversion is carried out by not all of the loop moving away. The termini of the loop remain in more or less the right position. But residues 250 to 250 to 260, I would say, these residues, they are displaced. So it's like a hinge-like motion by about yeah. So the closed one is where the loop is completely, it's the closest essentially to the active site. So we are measuring the minimum distance.
If I look from that chart,
Yeah.
it says 33.
It's not too large. It's not too large. It's a difference of between 31 to 35. Yeah. And only one small part of the loop actually moves out. So the N and C termini remain in the positions that they are, and a bit of it flips out. So you are absolutely right. It is not a huge movement, so to speak. But there is this very interesting interconversion. So it appears to move from open to closed to intermediate. And what is very interesting is we can capture these within the same structure. If we do not impose symmetry in the C1 structure, we can actually capture in different subunits, in different monomeric subunits, we can capture the open state, the closed state, and the intermediate state. So all of these three things can be captured in the same structure in a cryo-EM map of what we are seeing in dynamics. So essentially what we learned from here, the question is what is the new information that is coming from the native structure versus the recombinant structure? Well, what we found is that we can put some evidence to what was suggested about these enzymes about 10 years ago, that there would be some flexible loops which will control the entry of the substrate, and that we can now see. And then we can see that these can exist in different conformations which can block access to the active site. And we find almost an equal distribution of the three states, and primarily dependent on the hinge-like motion of the residues between 254 and 260, and because the distance that the loop is moving away is quite small. So this also can suggest why this enzyme gives preferential access to small peptides, like large protein molecules simply won't go in there. And we're still trying to work out the intricacies of the metal-dependent catalysis, because in the native structure, we are seeing that two zinc ions are there as opposed to the crystal structure. Yes. 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 Right.
Possible. From a study point of view, coordination between the units.
Yes.
Maybe one is in place.
Yes.
The other one might be different.
Yes.
Is it possible?
We should be able to do that. Yeah.
Does that be allowing to learn more about the?
Absolutely. In consort, what is happening? Yeah, absolutely. But the moment we apply symmetry, then we end up with just the intermediate state, and I think that is the case with the crystal structure, where if the loop density is not, if you average it, then even if the densities were there, in some places they would get averaged out.
But
so
you have individual particles.
We have individual particles. Yes, definitely. Right. Okay, so for the interest of time, I'm not going through the M18, where we are seeing, again, a similar kind of substrate association control by the presence of, yes, with symmetry. 3.4.
Distance.
Yes.
How the same range.
Yes.
All.
Yeah, that is, so we have done the MD with both the C1 as well as D3. And therefore, we think, so we've done this with the 2.75 as well as with the 3.4. And we are seeing a similar distribution. So that but yeah, that's a good point, that the resolution is not, really down to a very high point, but that we are not expecting, because it's very hard to reach there from this data.
Refinement.
Certainly, yeah, certainly it's feasible. Okay, so this is again with M18, we are getting more information from the native structure, and but these are all secreted proteins. So the next question we had was, well, what about the proteins that are retained in the merozoite membrane? Can we do anything about it? Now, if you know about membrane proteins, solving structures of membrane proteins is very, very challenging. When you purified them in homogeneous form, in extracted in detergent, even under those circumstances, it is very challenging. We don't know if this is going to give us anything. However, we extracted the protein from the membrane. So again, first isolation of the merozoite membrane, followed by detergent extraction. Now here we used different types of detergents in different combinations and concentrations to see what would give us the highest amount of extracted protein at high molecular weights. So we used DDM, LMNG, CHS, digitonin, CHAPS, etc., SDS, and visualize them in terms of the content and sizes. And then again, negative stain EM and cryo-EM. And so this is something that we are doing as of now. I'm just showing that one of the protein molecules, or one of the morphologies that looked really interesting, and it appeared that we had different orientations. So this is, we don't know what this is, but we can very clearly see protein density in between detergent micelle. And this is a movie. So the top one is in detergent micelle. Now, when we remove the micelle, this is what it looks like. So this again is a D2. So there is symmetry. There is a very clear D2 symmetry, and now we are again trying to identify using a combination of our knowledge about merozoite membrane proteins. Are there any proteins which are in D2 conformation? It appears that there are quite a few receptors which have D2 conformation. And the side chain density in some areas are clear enough, so that can help us identify what this membrane protein would be. Okay, so this was very highly heterogeneous sample from the component perspective. Very heterogeneous mixture of proteins. Now I come to the other problem, which can also be resolved to some extent using cryo-EM, which is, what if I have a component in different conformational states? Is it now possible to address separation? And this is something we are doing in the context of virus entry. So the step in virus entry that we are very interested in is the step called disassembly, which is where the virus has entered cells through receptor-mediated endocytosis, and now it has to fall apart within the endosome. So there are two concerted events. One, the virus capsid, which is very stable, has to fall apart to release the genome. And at the same time, the endosomal membrane needs to break so that the genome can be released into the cytosol for the next steps of replication, etc. So at this point, although cryoelectron tomography is available and people are getting excellent information out of tomography, however, the resolution is not good enough to follow individual virus particles at very high resolution to see what they look like as they release the genome. So what we tried to do to try to understand this conformational alteration in vitro was that we tried to work out a method to trigger disassembly in vitro. And this we do with the help of a process called DSC, which is differential scanning calorimetry. So we just give small packets of heat to the particle. And this came from a very old paper on poliovirus. When in 1975, people were trying to study how poliovirus falls apart. And they found that poliovirus has the same structural transition if it is incubated with receptor versus if you just have it in vitro and give it small amounts of heat in a very steady fashion. So the structural alterations are similar. And this just gave rise to the idea which prevails in the community, that virus particles, particularly non-enveloped viruses, which is just a protein shell with the genome inside, is essentially in a metastable state, and it is poised to disassemble, and it will disassemble once it gets the right conditions. And receptor binding is supposed to give it some energy, and that which can be replicated in vitro by just giving small quantities of heat so that it can go over the energy barrier and it can start disassembling. So we utilized DSC, and we found that we were working on this virus called flockhouse virus, which is an insect virus, and it's kind of like a model system for studying non-enveloped viruses. So we just subjected this to DSC, and what we found is that essentially there are two peaks in the DSC, which correspond to endothermic events. So there are two steps in which the virus appears to undergo significant conformational alteration, and so the conditions that a virus is being subjected to within a cell is receptor binding. Then there is low pH in the endosome. So both of these conditions are applied to viruses as they're falling apart. So we did these assays with and without application of low pH, and what we find is in low pH, the endothermic transitions that we see in DSC actually shift to lower temperatures. So it becomes easier in a sense. So this we have studied extensively. But because disassembly is not a synchronous or synchronized process, it's not something we can synchronize along all particles. When we take these particles from either of the peaks and we just put them on a cryo-EM grid and we're trying to see what are the different states, what we find again is a combination of different states. So if you just look at this negative stain image, this is not cryo-EM, this is stained with heavy metal. So we can see that these particles are clearly hexagonal and whole, and these are all icosahedral particles. And then you kind of see the particles getting rounder, and then you start seeing these openings, arising in the particle, and finally you appear to find a particle which is almost completely empty in the inside. So again, we went back and we took this heterogeneous mixture and we did cryoelectron microscopy. And then in the 2D state, we are trying to again separate out particles. So here, these are the rounder particles. These appear to be the more angled hexagonal particles, and these appear to be the empty particles. So this is just an example of the sorting, and I'm sure that in some of these sortings, we are mixing particles together because it's at the early intermediate state, it is very difficult to separate them out completely. But what we find is, so this is a reconstructed early intermediate. This is a late intermediate. We call it late intermediate because we are seeing something coming out of the particle. So it's in the process of releasing the RNA that is inside. In the early intermediate, what we see is that the particle expands asymmetrically. Although it is an icosahedral particle, the expansion is not happening across one axis. Overall, it is happening across one axis versus another. And if we look at this kind of expansion in a little bit more detail, I have 10 more minutes. Okay. So if we try to look at this expansion in a little more detail, we just have to take a quick look at virus symmetry parameters. So essentially, for this kind of virus, which is a T=3 icosahedral virus, we don't need to know all the details, but there are these 5-fold axis of symmetry. And then there is this 3-fold, and then there is this 2-fold axis of symmetry. And these are throughout the particle created by the protein molecules that make up the particle. So what we found was that the initial crack, as we would call it, happens at the 2-fold. So there are two types of 2-folds in this virus. So something called a flat contact and something called a bent contact. At the flat contact, we find RNA, which is genomic RNA, and we see the density for the RNA right where the protein, the protein-protein interaction site is. So essentially what we find is that the first opening leading to which will probably lead to the release of the genome, happens at the twofold, where we see a movement of almost, I would say, some 6-7 angstrom in the opening, but the 5-folds and the 3-folds remain more or less the same, except that there is also a little bit of change at the 5-fold, but not at all the 5-folds. So the 5-folds contain a density called the pocket factor. And this is there in a number of non-enveloped viruses. Nobody knows what the pocket factor is. It's just right there, and nobody has been able to identify it from the density, but it's right there at the 5-fold axis of symmetry. In the early intermediate, we see at least in half of the 5-folds, the pocket factor is not there. So the pocket factor has been released, but the opening is not broadened. The 3-fold remains more or less the same. The initial crack is at the 2-fold, and then we capture a state where the particle appears to be releasing something, which is probably the genome. And the release site is across from a 2-fold axis of symmetry, which tells us the release is probably through a 2-fold axis of symmetry. So we can see the internal density using cryo-EM, and we can reconstruct it. So when we look at the RNA density, what we find is that in the native particle, the RNA is present in 2 distinct layers. So this is the capsid protein, this is the outer layer of RNA, this is the inner layer of RNA. And in the early intermediate, the RNA seems to form like a complex. There are 2 strands of RNA in this particle, and they move away towards the inner edge of the protein shell, and this is probably preparation for it to be released. And also the RNA, if we look at what it interacts with, the RNA seems to interact with a small peptide which stays internal to the virus, and this is required for endosomal membrane penetration. So the release of the RNA and the release of the peptide for membrane disruption are probably coupled events. And finally, we reach a stage, and this is another, the last stage that we have reconstructed, which appears to be a completely empty particle. And here, instead of the particle just opening through, so what we find is, if we just look at which portion is missing, it's like 1 6-fold has completely moved away like a lid. So this is 1 hexameric component around the 3-fold, and this entire area appears to just move away like a lid, and then there is no genome inside the particle, and we don't see any RNA density whatsoever in this particle. So this was really interesting to us, and so we have an early intermediate, kind of mid intermediate, where the particle is releasing the genome, in the process of releasing the genome, and finally an empty particle where it has released the genome. I'm sure there are many states in between. Oh, these are just radially colored from the perspective of distance from the center. Yeah, so these are the tips of the capsid protein. So essentially there
was thing that has collapsed.
So this is a huge surprise to us, because all the literature on this particular type of viruses say that these are nodoviruses. They apparently cannot assemble without RNA. So we can get virus-like particles of these, but they will just take any RNA from the cytoplasm, and they would incorporate that, and so they require charge neutralization for protein assembly. But so the idea was that you simply can't find empty particles of nodoviruses. But we clearly can, and we can reconstruct this. And but the point here is there must be many stages in between, which we cannot identify or cannot separate out using cryo-EM, because we're dealing with a very heterogeneous mixture. So in terms of steps, what we think is happening is first the pocket factor leaves from the 5 folds, then we see these openings at the 2 folds, Then the genome starts coming out from one of the cracks in the two folds, and finally we have this entire area that just moves out like a lid to allow the genome to come out. But what has happened in between? And also why the two-fold? We don't have an answer for that. So again, we took the whole virus and we ran all atom empty simulation on the whole virus. Not very well, I think. There is a lot of things that we can do. But what we were trying to figure out is if we just draw, let's say, cylinders from the center of the particle towards all of the axes, like two-fold, three-fold, and five-fold, all the existing axes, and measure the amount of water molecules that are there throughout simulation time, can we figure out which are the paths, which are the more open paths, rather, where there's more water? That was our water movement, which is our only parameter that we utilized. And can we then say, which paths are more open towards genome release? And without going into too much detail, what we found was that the order of water release, or the order of free energy barrier, if we go from lowest to highest, the lowest barrier was at the quasi C2 and the C2, followed by C3, C5, quasi C3, C5, and then C3. And finally, the center of the capsid protein, where obviously there is the highest barrier because the protein is right there. Okay, so the question that comes to mind is then the C2 the weak link in all such viruses? Would the two-fold axis of symmetry be the one to crack in all of these viruses? So the answer to that, as far as we understand up to now, is probably no. We were studying flockhouse virus, and there were a couple of other groups which have reported similar structures from picornaviruses that I showed before, where the crack appears at the C2. So the idea was that two-fold would be the one to crack. But we are now working on another virus called feline calicivirus, which infects cats. This, again, is a sort of a surrogate for human noroviruses. And again, we triggered the similar method to find disassembly intermediates. Now here, when we look at the earliest disassembly intermediate, we find that the opening first occurs at the the quasi threefold axis of symmetry, not at the twofold. So the first opening is occurring at the quasi threefold axis of symmetry. So all we can say at this point is that genome release appears to be a directed process. It does not happen isotropically from all over the virus. There's some part of the virus that cracks first, a lid sort of comes out, and the genome is released. So it's a directed process. However, the RNA may have different exit routes, which would be informed by the type of, or rather the chemistry of the protein, specifically at the different symmetry axes. So I think my time is up. So in conclusion, we work on heterogeneous and dynamic protein complexes, heterogeneous from the perspective of just many different components mixed together, dynamic from the perspective of having different conformational states mixed up in the same sample. But what we are able to generate are dynamic snapshots of native states of proteins, which are very important if one is trying to develop inhibitors. A lot of our inhibitors fail when we try to convert them to drugs, and that there are many reasons for this, but one of the small reasons could be that we're working on recombinant protein, which is not giving us all the information that native protein might. And also in terms of vaccines, the malaria work, it has not been possible to develop a vaccine against malaria by working on many of the malarial surface proteins have high resolution structures available, and people have worked on those. But the idea is that the topology that is being presented to the immune system may be quite different on the surface of the malarial parasite. So we are hoping that the membrane components of the malarial parasite would give us a clue as to what exactly is the surface topology, which can then, the knowledge can be harnessed for generating better vaccines. And in terms of the virus disassembly work, this gives us some biological information some physical information about how viruses fall apart, disassembly is a very important step. So either hastening disassembly or stopping disassembly may be ways to develop inhibitors. So acknowledgements. So all of the work on malaria was done by a very brave postdoc in my lab called Devrat. And he is absolutely fearless and just goes into heterogeneous proteins like a warrior. And he attempted something that I, as a postdoc, would never be able to even try. Then Shubhamoy he did all the computational work. Pankhuri and Milan and Kirti worked on the FHV, the virus disassembly. And Gaurav also did the computational work on FHV disassembly. All the data was collected and analyzed at our cryo-EM facility and our HPC. And these are our collaborators and these are the funders. Thank you all for your attention.
Thank you so much for absolutely fantastic talk and the power of cryo-EM. Lots of hands. I'm sorry, I did not see the order they went up, and so I'll go with left to
Thanks. Fascinating talk, actually. Such beautiful images, actually, of these viruses. I just want to get back to the MD simulations of the viruses, and you got those free energies. I just want to get a sense of that. And what's the size of the system? That looks humongous.
Yeah, it is some 6 million atoms.
Wow. And that in solvent.
Yeah.
You actually put solvent around it.
Yes. So it took a long time.
Yeah, and but how did you get the free energies out? What the free energy barriers that you mentioned?
Yeah, so we are looking to see basically. So as I said, I mean, although we have all the trajectories as far as analysis goes, because nobody had done this before. How do you actually try to identify where the cracks are supposed to be? So we don't really have an answer for that. And the genome density, which we were showing in the cryo-EM structure, when we started the simulation, we only had the X-ray structure, where you can only see ordered RNA. So we don't see the bulk RNA. So there was nothing we could do with the bulk RNA. Now we have that information. So what we were trying to see essentially was, there are proteins like from the inner part of the capsid as we are looking out. So there would be protein barriers all over the place, right? So the idea is that if we can just find out in which direction, so water is going in, water is coming out. So in which direction do we see the maximum movement of water? And there can be, say, the barrier towards the protein barrier, so to speak, about the movement is the least. So this is what we are trying to quantify, again, not in a great way, and I'd be happy to discuss with you later. This is just something we came up with because there is no example in literature.
So just a physical barrier is what you're looking. Just sterics is what you're looking.
Exactly. But I'm sure there are better ways of doing it.
Excellent, beautiful images of capsids, especially the empty capsids. I have two short questions because you mentioned something about the water and calculations.
Yeah.
Are the viral particles are porous towards the water? The water can go in, come and...
It can. It absolutely can. Even small molecules can go in and come out. And we are actually utilizing this small molecule going in and coming out possibility to package sort of hydrophobic drugs in the virus, and we have been quite successful so far. But small molecules can go in and come out. And this was shown first in 2002, where people just put in a DNA intercalating agent, and it could go in and make the virus non-infectious. So the genome could not be replicated.
So very interesting. Yeah. I have another short question on the viral disassembly.
Yes.
Because disassembly happens in the late endosomes, I believe.
Yes.
And there are huge pH changes.
Yes.
So is it possible that somehow the C2 axis where actually the cracks are being opened?
Yeah.
Are there any protein residues which might be pH dependent so that you have huge changes in that?
question, but we tried to do this analysis with residues at C2, quasi C2, C3, C5, everywhere, and we don't see a distinction which would make the C2 the preferred destination. So we don't really know what that would be. But in terms of charge or in terms of pH-dependent residues, no, there are a couple of aromatic residues which are coming from the symmetry-related partner, and this is a phenylalanine. But other than that, I mean, there is nothing distinct, so to speak, which immediately comes to mind that this may be the controlling residue.
Beautiful. Thank you.
Thank you.
Yeah. Hello, ma'am. So nice talk.
Thank you.
So my question is that when we try to resolve a monomer
Yeah.
instead of a multimer,
Yeah.
of course you are gaining a resolution benefit. But my question is that doesn't it comes at a cost of losing resolution in the interfaces?
If
because-
we solve the monomer instead of the multimer.
Yeah. So why I'm saying is that in my experience, what I've seen is that it's generally whatever entity has been resolved, the resolutions, the local resolution is kind of like good towards the center and bad towards the periphery.
Yes.
Is it the case, right?
Yes.
So if you are if you are getting a monomeric unit, then clearly towards the periphery, it's going to be bad resolution.
Right.
And when you bring that monomers together to form multimeric
Yeah.
using symmetry operations,
Right.
so how trustworthy those?
So are you so monomeric and multimeric? What I'm getting is these are two completely different proteins that two completely different particles that we're talking about. Is that
No,
correct?
I am
Monomeric
saying-
particle.
I'm saying you are trying to capture the structure of a multimer.
Yeah.
But you are first getting the volume of the monomeric part, and then whatever structure you get from that, you apply symmetry operations, right?
No, we are getting the volume of the whole thing, except that we have not applied symmetry. So we are treating, so the volume remains the same, but we are treating it as, so we are treating it as a completely unique. So every part of it, we are treating it as unique. Versus when we apply symmetry, we are saying that 1/3 of the part is superimposable. So we are averaging the density. So therefore, if there is, let's say, two of the 1/3 look a particular way and one looks in a different way, you won't be able to capture the difference because you will average the density out. So when we don't apply symmetry, we are still looking at the full particle, except that every part of it we are treating as unique, versus in the symmetry applied part, we are saying that we will average, we have the capability to average.
Okay, yeah.
Fantastic work and such detailed analysis. Sometimes, that is underappreciated. So one of the things that I would like to kind of ask is this fundamental. There is one thing called compositional heterogeneity. Then you see and you pick particles and you see things are different.
Yeah.
These are different proteins. These are
Right.
But there is something called as heterogeneity of the intrinsic complex. For example, if you're looking at it, a particle.
Yes.
Now, if you have various conformational states, they are supposedly minima on
Yes.
some surface. Okay.
Yeah, yeah.
Now, can you sort of talk about the width of these minima? In the sense, you can also, because what you are doing is you're basically freezing out everything, right?
Yes.
So my question is that, can you talk about for one single particle,
Yes.
if you have done maybe 50 statistics
Yes.
out of 1,000,000,
Yes.
what would be the RMSD on those?
See, JD, that's a very good question, because we kind of think when we are doing structural biology, definitely X-ray crystallography, that the particle is existing in a very static state. The advantage of cryo-EM, I would say, is that if the particle was existing in 50 different states and we had only one particle, or even if we have multiple particles, perhaps some of the frozen states represent one of those 50. And it is possible to do, 3D variability analysis. So essentially, 2D variability, and you can also do variability analysis in 3D. So from the 2D of the same particle, one can generate several 3D structures, and this can be done quite efficiently to see what is the variability. And this comes out very nicely when you're looking at, maybe a protein binding to a substrate, or a small peptide bound, or an inhibitor bound, because you find different intermediate states. And this can be done quite efficiently in cryo-EM. Here, because we are dealing with so much heterogeneity to begin with, we think, we're, at least in the first part of the talk in the malaria, we are just happy that we are able to solve some structures. But in the conformational part, it is quite possible to work with different, or identify different states.
Okay, very, very futuristic question. So since you're able to look at such amazing heterogeneity, do you think in some foreseeable far future, we might be able to see protein structures within much more complex systems like cells?
Yes. I hope that will happen in my lifetime. But
when we think about the cell, there would be
Yes.
some proteins with larger copy numbers, some with lower copy numbers.
Yes.
And when you're looking at your entire grid, because I'm such an outsider, what is the kind of sample size, like in micrometers? How much of the area are you covering in general?
Right. So to answer your question, and this is something that is being done already. So this is a technique called cryoelectron tomography, and subtomogram averaging. So it is not at the resolution of single particle yet, and you can only look at very identifiable features. For example, so there are reports where people have done sectioned cells and identified specific protein complexes like the ribosome, for example. And then you can solve the structure of a ribosome, except the resolution is really terrible, but you can see it's a ribosome. So here, there's a lot of AI-based particle picking methods that are being developed, that if you know that this particular, or if you want to pick out a particular type of protein, if you already know the structure, you can maybe do some kind of AI-guided picking from cellular tomograms. So this is a possibility, but the resolution is still very low for all of these studies, and there's a lot of reason for it. I mean, everything from sectioning issues to electron damage to not being able to find orientations. There's a huge number of issues, but hopefully these can all be resolved at some point. So it's not too futuristic, maybe in the next 10 years.
Not very thank you.
Thank you very much.
Thank you very much. There is a tea served outside the West Canteen. Please can.
5:30.